Hi everyone, thanks for coming today. My name is Lynda Bradley and this is my asynchronous DEB 2021 Symposium talk. I'm a PhD candidate at Emory University and my primary advisor is David Civitello. My co-advisor is Lance Waller. Today I'm going to be talking to you about part of a chapter of my dissertation which aims to predict the effects of resource pulses on freshwater snail dynamics and the dynamics of the parasites that live inside them that can cause human disease. And of course I'm doing all this using a DEB-IBM. So resource pulses are a sudden, sharp increase in resource abundance and are ubiquitous in ecology across a variety of landscapes. Their causes are just as diverse as their location. Some are caused by regularly occurring natural events, such as mass cicada deaths where carcasses decompose, to anthropogenically caused pulses, such as agricultural runoff and sewage overflow. Resource pulse ecology also has important intersections with management and human needs, such as fish die-off events caused by algal blooms that impact human health and fisheries, and livestock polluting water bodies with their feces. So no matter how you look at it, they are pretty important to study. Resource pulses have cascading effects within ecosystems. When resources become abundant, this affects the population and community level dynamics. And I'm interested in exploring this scale crossing of ecological organization from the viewpoint of metabolic ecology. How does a resource pulse impact the individual level, and how does that scale up to the population and community level? So while I think I can learn a lot about the biology of different systems by doing this, I'm not just motivated by doing cool theory. Of course, I want to do cool and interesting theory, but I also want to do theory that can hopefully be readily used by applied researchers or managers who might need to answer questions about how a resource pulse will impact their system. So I'm going to show you a case study of how I'm investigating this. And it uses the Snail-Schistosome, don't say that 5 times real fast, the Snail-Schistosome system. So that system is composed of an aquatic parasite with a complex life cycle between snails and humans. Snails can be infected in freshwater systems, and then they eat food and produce cercariae, which is the portion of the life cycle responsible for causing human disease. Food is affected, of course, by the environment. Cattle feces, runoff, other forms of resource variation impact their food. And additionally, when snails are eating the resources, they can't instantly produce cercariae after becoming infected. It takes approximately 4 weeks for cercariae to develop inside snails before they are released. So using this system as a case study, one of my dissertation chapters investigates the question, when would be the worst time to have a resource pulse to the Snail-Schistosome system? And by worst, I mean when would we have the highest infection risk for humans as a result of a resource pulse? And this isn't just a theoretical problem. For example, invasive plant management can involve chopping up plants and throwing them into a lake, which provides a pulse of detritus for snails. Additionally, agricultural runoff caused by heavy rains has already been associated with increased schistosomiasis risk, and it's already been demonstrated in other snail trematode systems to directly increase cercariae production. So it might be useful to know when it's best to anticipate when a heavy rain will be the worst for infection risk. So if I were just to take resource pulse theory right off the shelf as it is, it assumes all individuals are equal and generally uses unstructured models that are disconnected from biological reality. But we already know that individual physiology and heterogeneous traits impact population dynamics significantly, yet physiology is still absent from current resource pulse theory. And normally I would spend time making the case as to why it's important to have physiology present, but as DEB fans, I know you're already a leg up in understanding that, so I'll give you the basic overview instead. So if I want to scale the individual response up to the population response and community level response, not all individuals are the same. They're not all equal, so a set of ODEs is never really appropriate for my work. Individuals are heterogeneous in their size, nutritional status, and other traits. So how would I predict a heterogeneous population of infected snails to respond to a resource pulse? For instance, how would starving juveniles respond versus well-fed adults? I might expect that the starving juveniles can utilize the resources to grow, whereas maybe the well-fed adults would lay a large clutch of eggs. And of course, history matters. Nutritional status is temporary, and so are resource pulses for that matter. It matters when you last ate, what you ate, when you were exposed to your parasite. All of these have characteristic times of responses, and the intersection of these with the timing of resource pulses determines the dynamics that we see at the population and community level. And of course, in between the time of consumption and utilization of resources, individuals store energy in reserve for use. So that differentially affects our starving juveniles versus our well-fed adults. And, these parts of biological reality are currently absent from resource pulse theory. However, luckily, I have a tool and you have a tool to begin answering these types of questions. DEB theory is a great way to do this. In the lab, our DEB model is basically a standard DEB model, but with parasites. So parasites can steal directly from reserves, and then they reproduce. And I'm sure this crowd won't be surprised to find that our DEB model actually predicts life history trajectories incredibly well for the system. For example, in this figure created by the lab, we see that the DEB model accurately predicts in the lines the actual cumulative cercariae produced in an experiment, which is the data points. And while this data was taken under constant food conditions, we already know from other lab work that these models also work under variable resource conditions. So that makes using them for my work pretty ideal. But again, I'm not just interested in modeling that individual level. Of course I need to, but I'm interested in modeling the population community level. So of course, I'm using a DEB-IBM. So the brief overview of our DEB-IBM is that we do it in R, and we have a simulated pool of 500 liters with 60 snails of a uniform size distribution from small to large. And then within the IBM, we have two other submodels built into it besides the individual host DEB model. So we have a between host model that calculates snail infections. So Myricedia are the life portion that infects snails. And then we also have the resource production model where we have logistically growing algae and detritus pulses, which I'll talk about more in a second. And then of course the DEB model. The good news about using this in this system is that we already know that these DEB-IBMs can generally predict seasonal patterns of actual empirical data, which is great for if you're trying to, not just cross scales, but cross, experimental to theory work. So the left panel depicts a different pond IBM simulation than I'll show today, and the right is a mesocosm data collected previously by my PI and a postdoc. And even though these are different environments, we see that the IBM can still capture this mid-seasonal characteristic peak of cercariae. So more reasons to be excited about using this. So here's the short of what I did. If you want to talk more like DEB method specifics, just reach out to me. I'm more than happy to talk all day about DEB stuff. But I think the most useful thing here is to just briefly explain how I manipulate the resource input. So I have logistically growing algae as a baseline, both in my control where there's no resource pulses and my experimental simulations where there are resource pulses. And then the pulses themselves occur at a weekly time point in the simulation. So one pulse at week 1, week 2, week 3, so on and so forth. The resource pulse is non-reproducible, which mimics things like, dead plants being dumped into a lake, die-off events, runoff, etc. And then I basically quantify how population of parasites respond to this, trying to answer the question, when is the worst for, pulse timing? So here are some of the results that I have so far. So pulse timing dictates the size and number of parasite peaks. So these are 5 simulation representative traces of cercarial density in red, and the black dotted line is the detritus input. So this is our no pulse control, and once again, we're seeing that characteristic mid-season cercarial peak that we see in mesocosms and other IBM simulations. And then if you pulse detritus in near the cercarial peak, this causes the peak to grow and also widen, which makes sense. And then if you pulse after the initial cercarial peak, you just get a small second peak. So I'm interested in the overall infection risk, which corresponds to the cumulative total cercarial density, not necessarily these temporal dynamics of cercariae. So how does overall infection risk trend with pulse timing? So to do that, once again, we look at the total seasonal cercariae, the infection risk under different conditions. So on the left panel, I have the no pulse control, and on the right panel, I have the data from doing detritus pulses at every week. And I see this really clear trend where pulses that occur mid-season increase infection risk greater than pulses occurring anywhere else in the season. But why? So, of course, turning to reserve density to answer this question. So this is the characteristic reserve density, once again, of 5 representative traces for our no pulse control. You can see that eventually mid-season, snails have a starvation event due to competition. So snails produce cercariae when they have a lot of energy on hand, and they stop when they don't have energy anymore. So that makes sense. And what we see is that when pulses occur before the snails start starving, this extends the period of time where they have high reserve density. And in the case of our third panel here, we can see that the pulse actually rescues a snail population trending towards starvation. And that's giving them basically time to rebound to really be able to sustain their infections. And if the pulse comes later in the transmission season, then many snails have already died, and the ones that remain are starving. So then the pulse just provides energy for a new cohort of snails, former cohort members that have survived but are starving, and many new starving juveniles. So this new cohort uses the pulse to rebound the reserves, which can facilitate shedding, but only once they have either had time to become infected or to rebound the reserves properly. So later in the season, the pulses are consumed by this new cohort. And earlier in the season, they're consumed by the old cohort before they starve. So then if we revisit this trend of infection risk, keeping that in mind, we see that if the pulse occurs early in the season, it fuels the growth of juveniles who are either not yet infected or can't shed very well because their parasites are not fully developed, or they're just not making as many cercariae per capita because they're smaller. So these snails are not developmentally ready to capitalize on the pulse. Whereas here in the purple, when a pulse occurs later in the season, it's utilized by the new cohort of snails after the original cohort has died. And furthermore, this cohort is made up of starving snails. So their nutritional status is so low, they already have lower per capita shedding. And the longer you wait for a pulse, the longer you have to wait for a pulse, the smaller and the more starved that cohort is. Whereas here in the middle of the season, the pulse occurs when snails are on their way to starving soon, but the pulse rescues them temporarily, giving them a longer time to shed cercariae. These snails are mature and their parasites are mature, so they make a lot of cercariae. I've also been looking at different components of the physiology to understand what mechanistic features drive these dynamics. And I'm also exploring more simulation sets across larger environmental parameter spaces, especially with regard to resource input. So one way, of course, I've been doing this is by looking at size distributions, and I just think this is interesting. So here we have a different set of simulations, but still the same DEB parameters, and we see that the relative density of the population remains basically as juveniles in the no pulse control. However, when I pulse in resources highlighted in red, I see that the juveniles are able to grow and eventually reproduce significantly, leading to a relative increase in juveniles again over a 6 to 7 week interval. So through all this, I have some take-home hypotheses. Pulsing in detritus near the middle of the transmission season is going to increase your risk the most. And high infection risk following a resource pulse occurs when snail populations are mature and do not suffer a mid-starvation event. Of course, I would like to cross back and forth between experimental and theory, so I'm testing these in a mesocosm right now, so fake little lakes basically in these tanks. And I'm also, like I mentioned, doing greater parameter space over environmental conditions and trying to understand where these trends hold true and where they might not, and doing formal statistics on these. I'm also investigating not just the quantity of pulses that get put into the system, but also the quality of these pulses. So thinking about nutrient availability, and of course using like multi-reserve DEB, more DEB to answer these questions. So maybe next time I do DEB stuff, I'll be able to talk to you about that. Thanks to everyone who is, doing this project with me, or is making life fun to do while I work on this project. So especially the Civitella lab, my primary lab, and my co-advisor Lance Waller. Shout out to my committee, and also of course my funding. If you have any questions or want to talk more, please reach out to me. I am more than happy to do that. Thanks for coming today.